So here is an analogy that might make it easier to understand how DNA makes RNA, how RNA makes protein. So we can think of a restaurant being like a cell, page 73, if you just came in. Now, in that restaurant, there is a back office, okay? There is a back office. And in that back office, there are cookbooks. And there is also a photocopier. Now here's the deal. In this particular restaurant, you are not allowed to take the cookbook out of the office. But what if you need a recipe in the kitchen? You can use the photocopier to make a copy of a recipe. And you can take the copy of the recipe and go to the kitchen. So now to make this attached to, I'll wait for you guys to finish writing and then we'll talk about what those all mean. So in our cell, in our restaurant, because the cell is the restaurant, yep. What's back office? The back office is like the nucleus. And what do the cookbooks represent? That's our DNA, because the DNA contains all the recipes. Okay? And we can use a photocopier to make a copy of the recipe. What would that copy of the recipe be? That would be the RNA. Specifically, what type of RNA? mRNA. So then you can take the copy and you can go to the kitchen. Where is the kitchen? The kitchen is the cytoplasm. So that's why RNA is necessary, because it's like you're not allowed to take the cookbook to the kitchen. So how are you going to get the recipe to the kitchen? You have to make a copy of it. That's your RNA copy that then goes to the kitchen. Making a photocopy. That's called transcription. Okay, so we've had our DNA, which is our cookbook, and the recipe itself, what's the recipe? The recipe is a gene. So there are many genes in the DNA. Just like there are many recipes in the cookbook. Now, the mRNA has a photocopied recipe. This is for a protein cake. Okay, there's the key. We're making some sort of protein, right? That's the recipe, is for making a protein cake. And each step of the recipe could be thought of as a codon. What is a codon? How many letters is a codon? It's a 3-letter code on what type of molecule? The mRNA. Now, who's going to do the cooking? Who's going to make this protein cake? That would be our chef, which is like the ribosome. Or a baker. Well, yeah. I think it's chef because in, have you guys seen South Park? I think they called it chef because they had a picture of chef. I don't know. I stole this from somewhere. I'm like, somebody made some good pictures. Excellent. And then we have the tRNA, which is like the kitchen hand, and it brings things to the chef. And the amino acids are the ingredients. So when you have all of those things together, you can make the food, which is the protein. Okay? So if that's helpful, that's wonderful. If it's not helpful, you can just be like, okay, I understood it anyway, or I didn't understand it and I need to do a little more work on it. Okay? So that is our little analogy for making the DNA. And the making of the food is called the translation. That's where we take the mRNA code and we make protein. Okay, I can't remember if this is a very good sheet or not. I'm just going to give it a try. Okay, let's see. We've got, yeah, this one here. Yeah, no, I don't really like that one too much. Let's see if this one's better. Oh, here we go. I don't really love this one either. You know what I'm going to do? If you would like to get yourself just a piece of paper, we're going to draw stuff together. Okay? And if there's anyone who needs a copy of this when we're done, just let me know. Okay? And remember, there's paper at the back if you want. See if you can remember. Yes. So draw and then talk, or write and then talk?
It's not.
Oh, okay.
Yeah. I know that's hard.
Okay. Okay. Yeah. The other thing is I'm going to print this out. So if she just wants to sit and listen, I can print this out at the end. Yeah? Okay, okay, yeah. And that goes for anyone. If you want a copy of this at the end, I can print it out for you. So if you're like, it's too hard to keep up with the writing and the drawing, it's okay. Don't worry about it. So we had some vocabulary, and we had a new vocabulary word yesterday that started with an H and also started with a CH. See if you, discussing with your partner, can remember what that vocabulary word is. So we know it's homo, homologous. And they are homologous chromosomes. Whoops, I've forgotten O. Homologous chromosomes, how many of them are there? Like for a single chromosome? Like chromosome number 1 has how many homologous chromosomes in a cell? I'm asking the question really, really badly. They always come in. So they always come in pairs. One of the chromosomes came from mom. And one came from dad. Do you remember the symbols? One's a shield and spear. Okay, so that's the shield and the spear. The other one is a hand mirror. I'm not quite sure why it has a little, thing on it, but it's like a little plus. It's like a little person, I guess. So these chromosomes two things about them are always the same. They're always the same size, and they're the same shape. And they have, actually, it should be the same genes, so I'll have to put that. The same genes in the same places or locations. So if we were to draw a really simplified version of this chromosome, often we will draw it just like a sausage, okay? Like a big oblong shape, okay? Now there's these things somewhere in the middle of these chromosomes that we have not yet given a name to. And today we will give them a name. That is called a centromere. And it's not really important for chromosomes in this state. It's more important once they have replicated. What does it mean for a chromosome to replicate? We make a copy, we make an exact copy. This one, we're going to have this be the thumb gene. And we're going to represent it with a big letter T. Now we're going to see how badly I can draw. That's a hitchhiker's thumb, okay? I'm really sorry that it does not look like, it looks like a turkey or something. That's supposed to be a thumb. And it bends, it's supposed to bend more. Okay, now it looks like a teapot. I don't know, it's supposed to be a hitchhiker's thumb. Now, over on our other chromosome, what's going to be in that same location? That's going to be a thumb gene. And this time we're going to give it, this person, a little t. Okay, and here we now have a fist. And here's the thumb. Okay, they've got a really long thumb. I don't know what's wrong with this person, but it doesn't go. This is a straight thumb. It's like if they're holding a fist like this. I don't know. These chromosomes are the same So we're not putting anything new here. This is just review of what we just said. Yep, these chromosomes are the same size. They are the same shape. And they have the thumb gene in the same place. We could draw a whole bunch more genes in different places, but they would all be in the same place for each one. So maybe up top, maybe we have an earlobe gene. Yeah, of course. Yeah. And for this one, I'm going to say we have the earlobe gene, little e, and the earlobe gene, little e. And the earlobe gene, little e, is an attached earlobe. So if here's your head and here's your ear, it just goes straight into your head, okay? So this person is big T, little t, for the thumb. And this person is little e, for the earlobe. There's two words that start with H. One is if both copies of the gene are the same, and one is if the two copies of the gene are different. Now, probably you can remember at least the first part of this word. So if we have two copies that are different, what's the first part of the word? Hetero. And what are the next two letters? ZY. So we have heterozy, and we have for the one they're the same, it's going to be homozy. And then what goes at the end of the word? Gous and Gous. If you just put a U there, I'm not going to get worried. Okay, your spelling is not super important as long as you're close. So we have two that are different. That is heterozygous. And we have two copies that are the same, which would be homologous. So we could say TT is heterozygous for the thumb trait. And E is Homo, and then Zy. What comes after Zy? Gus. Homozygous for the earlobe trait. T and T are different somethings of the same gene. Alleles. Allele. Alle, 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 alle. Now I'm just being silly. Okay, alleles. But if you want to call them alleles and that helps you spell it, you go for it. Okay? Ms. Lamb, she's one of our biology teachers. She's like, I don't know what's with my science students. They're all calling alleles alleles. And I'm like, I'm sorry, that's my fault. Yeah, well, exactly, because it is kind of a weird word to spell, right? How do you spell allele? That's not a normal sort of a word, right? So allele sounds like something Hawaiian almost. Allele, you know? Dream of tropical islands, allele, yeah. How are we feeling about all those new vocab words? Are they starting to stick a little better? Okay. So if I said Homo, there's two endings you could put on that thing for our new vocab words. What's one of the things you could put on Homo? Zygous, or you could say homologous, and homologous goes with chromosomes. And then if we say hetero, the ending would be zygous. Okay. And different versions of the same gene are called alleles. Alleles. Okay. Ooh, and what's the new one? In the center of the chromosome, there's this sort of like oval-shaped blobby thing. What's that called? Ah, no, in the middle of the chromosome. Yeah, it's a centromere. We haven't talked about that one much yet, but so we'll deal with it today and tomorrow a little bit more, because we're going to now look at what happens when a cell divides. And when we say a cell is dividing, we mean it's making an exact copy of itself. We're going to do like a little cell division with the most simple cell possible. We're just going to have 2 chromosomes, okay? We're not going to try and have how many like a person? 46. We're not going to try and have 46 like a person. We're just going to draw 2 because it makes our life really easy, okay? Now, can you imagine, now over here, you might not want to draw this because this part is wrong, okay? This is wrong. So if we had a chromosome, starts out as, this part's correct. It starts out as, what form is that of our DNA? That is chromatin. That's our nucleus. Now let's imagine that this thing condensed, okay? The chromosome condensed. And we had 1 chromosome from mom, and we had 1 chromosome from dad. Is this going to work? Am I going to be able to split the cell in half and have identical daughter cells? No. Why can't I split this cell in half right now? Like if I was to split it in half, what would you end up with? You'd end up with a chromosome, whoops, that went weird. You would end up with the chromosome from dad here and the chromosome from mom here. Then what if you wanted new cells, right? Because you don't just stop at 1 division. What are you even going to do with this? Right? Like there's like, you can't, there's like, do you have a half a chromosome? Right? Like how does that work? If you start splitting things in half, you would end up with a half of the DNA and then a quarter of the DNA and then an eighth of the DNA. See how that's a problem, right? Like things aren't going to be able to work. So before the cell divides, the DNA needs to make a copy of itself. And what's a word that we could use that means to make a copy of itself? Replicate. And it's really hard to show replication of chromatin. because chromatin is long and noodly and messy. Messy, messy, messy. So we're not going to show replication in chromatin form. We're going to show it like this. So here's our cell. Here's our nucleus. We have one chromosome from dad, one chromosome from mom, and remember what's that they have in the center there? Centromere. Okay, so they have centromeres, each of them. And so what we will end up with then is we will end up, what will the chromosomes look like now? Instead of looking like little sausages, they will look like little X's. Okay, so we started out with unsomething homologous chromosomes, unreplicated. Ah, why are you buzzing at me? It says, let's get moving. Well, thank you, Watch. I'm teaching. I cannot do that. And we finish with what do we finish with? Replicated. Homologous chromosomes. And do you remember what these little doodads here are called? That is it's in the center of the chromosome. Centromere is in the center of the chromosome. Yeah, for sure. And it holds the two halves together. And Kita, you have a question? Yes. Yeah, when we have the replicated chromosomes, the two halves are called sister chromatids. We're going to draw that just down below so that we have that labeled correctly, okay? So The centromere is in the center of the chromosome, and it holds the two sister chromatids together. The centromere holds the two sister chromatids, which we'll label again in just a moment, together. Yes, of course. Two sister chromatids make up a replicated chromosome. Now, sometimes people get mixed up between homologous chromosomes and sister chromatids. Okay, so homologous chromosomes they're not attached to each other. And they have similar, yeah, please feel free to get yourself more paper if you need it. They have similar DNA, but not exactly the same. On the other hand, we could have sister chromatids Are they attached to each other? Yes, they are attached to each other. What are they attached to each other by? the centromere. Is the DNA similar but different, but a little bit different, or is it identical? The DNA is identical. Now we're going to simplify our drawing a little bit, okay? So we're going to start with, we're going to make, instead, our unreplicated chromosomes are going to look like this, because that's easier to draw than a blobby sort of thing. And our replicated chromosomes are going to look like this, because again, that's easier to draw, okay? The nucleus is really big here because we need space to draw, right? So normally the nucleus would not take up that much of the cell. We're ignoring the chromatin because chromatin is too hard to draw like actual seeing things. What would happen next? What do we have in that first nucleus? In that first nucleus, we have unreplicated chromosomes. We have a homologous pair. What has to happen next before the cell divides? They don't line up yet. They have to replicate. And if you're using just one color pen, you can do like, dashes for one of them to make it look a little bit different. You could have like one that's dashed and one that's like full lines. Okay, if you've only got like, say, a pencil or something. Now, you're absolutely right. After they have replicated, what do? They line up. So the DNA replicates. The chromosomes line up. in the middle. That's too small a circle. There we go. Now what happens? You've got these things, these X shapes in the middle. Gesundheit. So you have a chromosome with two sister chromatids, and in order for us to get the genetic material, each identical copy, here I'll make them identical copies, there we go, identical copies to opposite ends of the cell, what do those two sister chromatids have to do? They have to separate. So they're actually going to pull apart. So everyone do the sister chromatid separation dance with me. Woo! 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 Okay, so we're going to watch them as they get pulled apart. There's actually little strings almost in the cell that attach to them and pull them apart. So one is getting pulled this way, and one is getting pulled that way. One is getting pulled this way, and one is getting pulled that way. So the sister chromatids are pulled apart. Oh, and actually in the meantime, you know what has actually happened? The nuclear membrane has disappeared. It's going to make life a lot easier. That actually happens earlier, but we're not going to worry about it too much. I mean, honestly, you don't need the major details of this. Like, you just kind of need to know that this is why a cell divides. This is how a cell divides. I'm not going to make you like, oh, please draw me the steps of cell division. I will not do that. I might have like at a certain stage, I'd be like, what do they do next? Do they A, line up? Do they B, replicate? You know what I mean? Like it'll be like a choice kind of a thing. Yeah, on a test, yeah. Okay. And then once they get to opposite sides of the cell, actually, maybe I'll let you finish writing first. And if you need more paper, don't be shy about getting more paper. So what's going to start happening now is that we now have the sister chromatids are at opposite ends of the cell, right? They pulled apart, and now they are at opposite ends of the cell, and then the cell starts to pinch in the middle. cell membrane starts to pinch the cell in two. And can you imagine what happens next? They actually literally, like, it starts to pinch them in two, it starts to pinch them in two, and then it finally gets to the point where it, two new cells. So you could do this little dance with me too. You could be like, the cell starts to pinch, and eventually the two membranes fuse together, and then two new cells. What do we call those two new cells? Are they son cells? No, they're daughter cells. Okay. Is good? Okay. Don't worry, I'll put it back up in just 1 second. Anyone else who wants a copy of this? One, two, okay, like about maybe 15 of you? Okay, you know what, I'll make one for the entire class. It actually might take me just a moment to do, so I'll let you finish copying this and then I'll make it for you. If it's not done by the end of, when is class over? 10:20? Oh yeah, I'll have time, okay. So it should be okay. So what's your homework tonight? Go work on your cheat sheet. You've now got like some really good notes that you could like, you know, take stuff and put on there. And if you've taken the notes and you just want to cut it out and paste it on, you could do that, but you will get more out of it if you write it or draw it a second time, okay? Okay. Yes. Oh, okay. I'm not sure there's a lot I can do about that, but we'll see. Yeah, okay.